For an individual weak acid or weak base component, see buffering agent. For uses not related to acid-base chemistry, see buffer. A buffer is an aqueous solution consisting of a mixture of a weak acid and its conjugate base, or vice versa. Its pH changes very little when a small amount of strong acid or base is added to it, and thus it is used to prevent changes in the pH of a solution. Buffer solutions are used as a means of keeping pH at a nearly constant value in a wide variety of chemical applications. Many life forms thrive only in a relatively small pH range, so they utilize a buffer solution to maintain a constant pH. One example of a buffer solution found in nature is blood. Principles of buffering. Buffer solutions achieve their resistance to pH change because of the presence of an equilibrium between the acid HA and its conjugate base A. HA++A. When some strong acid is added to an equilibrium mixture of the weak acid and its conjugate base, the equilibrium is shifted to the left, in accordance with Le Chatelier's principle. Because of this, the hydrogen ion concentration increases by less than the amount expected for the quantity of strong acid added. Similarly, if strong alkali is added to the mixture, the hydrogen ion concentration decreases by less than the amount expected for the quantity of alkali added. The effect is illustrated by the simulated titration of a weak acid with pKa equals 4.7. The relative concentration of undissociated acid is shown in blue and of its conjugate base in red. The pH changes relatively slowly in the buffer region, pH equals pKa plus or minus 1, centered at pH equals 4.7 where HA equals A. The hydrogen ion concentration decreases by less than the amount expected because most of the added hydroxide ion is consumed in the reaction, OH plus HAlH2O plus A, and only a little is consumed in the neutralization reaction, which results in an increase in pH, OH plus H plus H2O. Once the acid is more than 95% deprotonated, the pH rises rapidly because most of the added alkali is consumed in the neutralization reaction. Applications. Buffer solutions are necessary to keep the correct pH for enzymes in many organisms to work. Many enzymes work only under very precise conditions. If the pH moves outside of a narrow range, the enzymes slow or stop working and can denature. In many cases, denaturation can permanently disable their catalytic activity. A buffer of carbonic acid and bicarbonate is present in blood plasma, to maintain a pH between 7.35 and 7.45. Industrially, buffer solutions are used in fermentation processes and in setting the correct conditions for dyes used in coloring fabrics. They are also used in chemical analysis and calibration of pH meters. The majority of biological samples that are used in research are made in buffers, especially phosphate-buffered saline at penthouse 700. Simple buffering agents. For buffers in acid regions, the pH may be adjusted to a desired value by adding a strong acid such as hydrochloric acid to the buffering agent. For alkaline buffers, a strong base such as sodium hydroxide may be added. Alternatively, a buffer mixture can be made from a mixture of an acid and its conjugate base. For example, an acetate buffer can be made from a mixture of acetic acid and sodium acetate. Similarly, an alkaline buffer can be made from a mixture of the base and its conjugate acid. Universal buffer mixtures, by combining substances with pKa values differing by only 2 or less and adjusting the pH, a wide range of buffers can be obtained. Citric acid is a useful component of a buffer mixture because it has 3 pKa values, separated by less than 2. The buffer range can be extended by adding other buffering agents. The following 2-component mixtures have a buffer range of penthouse 3 to 8. A mixture containing citric acid, potassium dihydrogen phosphate, boric acid, and diethyl barbituric acid can be made to cover the pH range 2.6 to 12. Other universal buffers are Carmody buffer and Britton-Robinson buffer, developed in 1931. Common buffer compounds used in biology. Biological buffers cover 1.9 to 11 pH range. Buffer capacity. Buffer capacity, I², is a quantitative measure of the resistance of the buffer solution to pH change on addition of hydroxide ions. It can be defined as follows. where Dn is an infinitesimal amount of added base and D, P, H+ is the resulting infinitesimal change in the cologarithm of the hydrogen ion concentration. With this definition, the buffer capacity of a weak acid, with a dissociation constant Ca, can be expressed as where Ca is the analytical concentration of the acid. pH is defined as log 10, H+. There are three regions of high buffer capacity. At very low pH, the first term predominates and I² increases in proportion to the hydrogen ion concentration. This is independent of the presence or absence of buffering agents and applies to all solvents. In the region P, H+ equals pKa plus or minus 2. The second term becomes important. Buffer capacity is proportional to the concentration of the buffering agent, Ca, so dilute solutions have little buffer capacity. At very high pH, the third term predominates and I² increases in proportion to the hydroxide ion concentration. This is due to the self-ionization of water and is independent of the presence or absence of buffering agents. The buffer capacity of a buffering agent is at a maximum when pH+ equals pKa. It falls to 33% of the maximum value at pH+ equals pKa plus or minus 1 and to 10% at pH+ equals pKa plus or minus 1.5. For this reason the useful range is approximately pKa plus or minus 1. Calculating buffer pH, monoprotic acids. First write down the equilibrium expression. Ha plus H+, this shows that when the acid dissociates equal amounts of hydrogen ion and anion are produced. The equilibrium concentrations of these three components can be calculated in an ICE table. The first row, labeled I, lists the initial conditions. The concentration of acid is CO, initially undissociated, so the concentrations of A and H+ would be 0. Y is the initial concentration of added strong acid, such as hydrochloric acid. If strong alkali, such as sodium hydroxide, is added, Y will have a negative sign because alkali removes hydrogen ions from the solution. The second row, labeled C for change, specifies the changes that occur when the acid dissociates. The acid concentration decreases by an amount X and the concentrations of A and H+ both increase by an amount plus X. This follows from the equilibrium expression. The third row, labeled E for equilibrium concentrations, adds together the first two rows and shows the concentrations at equilibrium. To find X, use the formula for the equilibrium constant in terms of concentrations. Substitute the concentrations with the values found in the last row of the ICE table. Simplify to. With specific values for CO, Ca and Y, this equation can be solved for X. Assuming that pH equals log 10, H+, the pH can be calculated as pH equals log 10, X plus Y. Polyprotic acids. Polyprotic acids are acids that can lose more than one proton. The constant for dissociation of the first proton may be denoted as Ca1 and the constants for dissociation of successive protons as Ca2, etc. Citric acid, H3A, is an example of a polyprotic acid as it can lose 3 protons. When the difference between successive pK values is less than about 3, there is overlap between the pH range of existence of the species and equilibrium. The smaller the difference, the more the overlap. In the case of citric acid, the overlap is extensive and solutions of citric acid are buffered over the whole range of penthouse 200 to 7.5. Calculation of the pH with a polyprotic acid requires a speciation calculation to be performed. In the case of citric acid, this entails the solution of the two equations of mass balance. Ca is the analytical concentration of the acid, Ch is the analytical concentration of added hydrogen ions, I²Q are the cumulative association constants. Kw is the constant for self-ionization of water. There are two nonlinear simultaneous equations in two unknown quantities, A3 and H+. Many computer programs are available to do this calculation. The speciation diagram for citric acid was produced with the program HIS. In general, the two mass balance equations can be written as. In this general expression, A stands for the concentration of the fully deprotonated acid, and the electrical charge on this species is not specified. See also, Henderson-Euro-Hasselbalch equation, buffering agent, goods buffers, common ion effect, metal ion buffer, mineral redox buffer. References. External links, online pH buffer calculator, phosphate buffer.